Hi, today I'd like to go over gene regulation and mutation notes. This sheet can be found in packet page 56, so page 56 of your packet, and eventually it will go on page 57 of your notebook. Okay, so when we talk about gene regulation, we're talking about how genes are turned on or turned off, or another way to say that is that they're being expressed or not expressed. So let's jump right in. In gene expression, again, certain parts of DNA can be turned on or turned off. Turned on or turned off. If a gene is turned on, it means it's making that protein. So, for example, a turned on gene, what does that mean? It means that the gene is being expressed. That's kind of the fancy science way to say that. And that means it is making that protein. Okay, so again, if another way to say that a gene is turned on is to say that it is expressed. Again, a turned off gene is not expressed. Which means it is not making that protein. That particular protein, whatever that protein might be. Now, sometimes things can go wrong. And when things go wrong, that is normally not a good thing, but it doesn't always have to be bad. What I'm talking about are mutations. When we typically hear the word mutation, we usually think of bad things. And yes, bad things can happen, like cancer, from mutations, but not all mutations are harmful. Some mutations can be beneficial, and actually some mutations can be what we call silent, which means that they don't have an effect on the organism at all. So it really just depends. But our definition of a mutation is any change in the sequence of nitrogen bases in DNA. So any change in the sequence of nitrogen bases in DNA. So for example, let's say I've got a sequence of DNA, and I'm not going to draw all the sugar and phosphates, but let's say I've got a sequence of DNA T, G, A, T, A. Of course, now you know your base pairing rule. The other side obviously would be A, C, T, A, T, because remember DNA is double-stranded. And of course, we've got our hydrogen bonds in between our nitrogen bases. So what we're talking about is a mutation is when, and it can occur in different ways, but for example, one mutation would be when a nitrogen base change. So for example, it's supposed to be a cytosine there. Well, what if the cytosine is not there and instead there's a thymine? Okay, a thymine took its place. That would be considered a mutation. That would be considered a mutation because, again, it's a change in the sequence of nitrogen bases. So the order of nitrogen bases is not correct, is not what it should be. Now, this is just one type of mutation. There are other types that we're going to go over. So anything that causes a mutation is going to be called a mutagen. A mutagen is some factor that can cause mutations. And let's go over some examples. Sometimes mutations just happen randomly. There's, spontaneously. In other words, there wasn't an environmental factor that caused them. So sometimes there's just random mistakes that can happen when DNA maybe is replicating. And again, sometimes just a mistake happens. There's supposed to be a T there, but it's a C. There's supposed to be an A there, but it's a G. Now, DNA does have a way to proofread itself. It almost has a built-in spell checker. We're going to talk about that in just a few minutes. So sometimes the mistakes are just random. We call these spontaneous mutations. Another cause of mutations is radiation. For example, UV light. This is one reason that you're supposed to wear sunscreen, to prevent UV rays from damaging your skin. Well, particularly what we're talking about is your DNA. You know that excessive sun exposure can lead to skin cancer or even x-rays. This is why they put that heavy jacket over you when you're getting a certain part of your body x-rayed, to protect those other areas of your body from being exposed to the x-ray. So when you go in for example, a dental x-ray, the dentist will put that jacket over you, that lead jacket, to prevent the x-rays from getting to some of your organs. Okay, obviously we just need to expose your teeth to those x-rays. Another cause of mutations are certain chemicals, like formaldehyde or petroleum ether, or some of the chemicals in tar. Obviously that's going to be related to smoking, and obviously related to lung cancer. And then some viruses can cause mutations. The best example I can give you here is the HPV virus, the human papillovirus, which can cause cervical cancer. Of course, there is a vaccine, an HPV vaccine that is given to girls, of course, and boys, because of course it can be passed on to girls from boys during sexual intercourse, and obviously you want to have that protection for girls. So it is offered to boys also. It is something that is recommended. Can we inherit mutations? Yes, absolutely. So reproductive cells, these are your gametes. These are your egg and your sperm cells. So reproductive cell mutations. Now, the fancy way to say that are germ cell mutations. Germ cells are egg and sperm. So sometimes these are called germ cell mutations. Become part of the future offspring's DNA. So you're passing the mutation on to your offspring. So when the mutation is in the egg or the sperm, it's not you that's affected, it's your offspring that's going to be affected by the mutation. Okay, so these are inherited. So if the mutation is in your egg and sperm, then that is going to be what we call an inherited mutation. So they are inherited. Again, if it's in egg and sperm. But sometimes mutations don't happen in egg and sperm. Sometimes they can happen in your body cells, like your skin cell or your lung cell or your pancreas or liver cell. So body cell mutations are not passed on to offspring, so they're not inherited, but can cause problems for the individual who has the body cell mutation. And of course, we have a fancy way of saying body cell mutations, that those can be called somatic cell mutations. Somatic cells are body cells, like I said, skin cells, pancreas cells, liver cells, lung cells. Those are all part of your body. So they're called somatic cell mutations. Can we prevent random mutations? Okay, that's going back up here. So when DNA is copying itself, mistakes can happen. Just like if you're typing something on the computer, you can actually hit, the wrong letter. Okay, well, there's a built-in spell checker, if you will. And there's an enzyme called DNA polymerase. There it is. It ends in ACE. Okay, remember, enzymes end in ACE. It is an enzyme that will proofread the DNA, and it will actually correct the problem. So it can, if it sees that there's a wrong nitrogen base, it can actually cut out the nitrogen base and insert the correct one. So it can proofread and correct mistakes in the DNA. Sometimes it can't catch all of them, but it can catch a lot. Now, what are some of the types of mutations? Okay, well, the first type of mutation is going to be something called a point mutation. This is when one nitrogen base is switched out, or obviously substituted. When you switch something out, it's substituted for a different nitrogen base. Okay, so for example, let's look at our original DNA. Okay, this is the correct sequence. This is what it should be. So it should go in the sequence AAG, TGC, TAG, CCG, AAC. But what if there's a point mutation that happens? A point mutation. Okay, well, let's see what's wrong. Again, it should be there it is. Okay, so right here is our point mutation. Right here is our point mutation. It should be a G, but it's now a C. Now the rest of the letters are right. TGC, TAG, CCG, AAC. So right there, it should be a G, but it's now a C. So the C was substituted for the G. So that is considered a point mutation. A point mutation. Our next type of mutation is going to be something called a frame shift mutation. This is caused by an insertion. So you're going to add, okay, insert an extra nitrogen base or delete a nitrogen base. And that's going to cause what we call the reading frame to be shifted over. So for example, here's our normal DNA, and we got AAG, TGC, and then look, we have difference. It should be TAG. Well, I've got TAG here, but you can see that an A has been inserted. That's going to cause the reading frame, which codes for an amino acid, to be off one. So it should read TAG, but it's not, and that's going to code for a particular amino acid. It's ATA. Well, the next amino acid is the next codon is going to be CCG. Well, that's not it because everything's shifted over one. It's GCC, et cetera, et cetera. So all of these amino acids are going to be wrong. Whereas in the point mutation, only one amino acid would be altered, only one. So a frame shift mutation has the capability to cause a lot more, I guess, damage because several amino acids are going to be altered. And then the last type of mutation is a chromosomal mutation. And we'll talk more about that later when we get to cell division and genetic diseases. So again, here we got the original chromosome. And we can see that this piece right here